Hello and welcome to our bladder cancer live buzz around session from the Uromigos 22 live meeting in Nashville. You'll see that each panel member is given a buzzer. They are shown a slide that they've not seen before, and whoever buzzes in first from the panel then talks for about 1.5 minutes on the question. There's then a bit of a discussion, and if deemed adequate by the rest of the panel, the person who answered the question is then allowed to leave the competition and doesn't have to answer any further questions. That eventually ultimately ends up with one panel member remaining, or indeed unless we run out of questions first. It's possible that one of the panel members can appeal or disagree with the original person who answered the question, and that can bring that person back into the competition. So it's possible to be eliminated and be brought back into the competition. And I hope that makes it more fun and not too confusing. I hope you enjoy the session.
Okay, so this is the first slide. This is not a warm-up. There's no warm-up around here. It's to whoever goes first. 5, 4, 3, 2, 1.
Wow. So I'm describing the slide?
You know, you've got to describe, not describe the slide, you've got to talk about the question. So the question is, what's the future of erdafitinib in urothelial cancer? And this slide is the Norse trial. Just to summarize the story quickly, in those individuals who have FGFR abnormalities, we know that they seem to do better, or they respond to therapy. And Petros, over to you.
Number one, it's very difficult to do these trials because of the biomarker testing requirement. And that, I think, may be a barrier to do a phase 3 trial. To me, when you showed the data, Tom, the Norse trial, I was very impressed about the response rate. Small numbers, of course, underpowered 99 patients in each arm or so, but the response rate was compelling enough for a phase 3 trial. I assume the biomarker testing may be a limitation. Erdafitinib has an established role in metastatic urothelial cancer. It's not being used that much because of biomarker testing. I see the role of erdafitinib further, potentially in early disease stages when the target is more frequently expressed, but the issue will be that clinical trials will be compared.
I decided I'm going to buzz him at 30 seconds.
Okay. So, Petros, you've got 30 seconds left.
No, he stops at 30 seconds.
Stops!
He only has 30 seconds.
Okay. So anyone to follow up on Jonathan said, what Petros has said?
I think that FGFR inhibitors in combination with checkpoints are probably going to play a role in the future of bladder cancer treatment. And based on the regorafenib-atezo data, based on the erdafitinib-citrilimab data, there's something, you're probably taking cold tumors and making them hot. We don't have a ton of supportive data yet in humans, but I think this is real.
And what trials do we have that are going to support that beyond the Norse trial?
At the moment, none that I know of. Maybe there's a checkpoint plus intravesical delivery. I don't know, actually.
So we have a trial coming in the neoadjuvant setting of erda plus atezo. So that might be interesting up front.
Okay, that's good. So now we're going to have
What's going on with the cards? I don't understand.
No, now the audience has to vote about whether Petros can be eliminated or not. Because if they give him a red, unfortunately, he's back in the competition. So let's have a vote for Petros right now. We've got green.
No,
We've
green
got
is good.
Green's good. Green's good. Green's he's eliminated. Yeah. So everyone's green. So Petros, you're good to go. It was a green. So let's move to the next question. Are we ready? Everyone's fingers on buzzers, if we may.
Hold on. Okay, ready.
You ready, Brian?
Petros is out.
Petros is now out of the competition.
I'm not using any buzzer.
Fingers on buzzers, everyone. Okay, 5, 4, 3, 2, 1.
Jonathan.
So this is a study I did. Took me a minute to recognize the slide. Durvalumab versus nivolumab. So PARP inhibitors have been tested in bladder cancer in several trials. Unselected seems to do nothing. Selected patients, seems like there's something there. Is there really a benefit in combination with immunotherapy? It's an open question. I think there is a story here, but it's a relatively small patient population. This was a platinum-ineligible trial. Also, defined pretty specifically based on the Galsky criteria plus one other. And so this is, I think we need to move forward either with the current generation of PARP inhibitors or the next generation of PARP inhibitors, which are potentially more effective.
Jonathan, 2 questions from me. Firstly, what was the role of the biomarker in this setting, and is the biomarker reliable?
So the biomarker I think is reliable. It's Foundation Medicine based HR mutations, homologous recombination repair mutations. The role of the biomarker, the study was initially designed for, pre-specified for all patients. There wasn't an appetite to wait as long as it would take to identify all those patients. So it was a pre-specified secondary endpoint to look at this patient population. And so this is not the primary endpoint of the study, this is the secondary endpoint of the study, but it is pre-specified in the statistical analysis plan.
Jonathan, the control arm, the durvalumab, the PFS it crashes like a stone.
Yes.
What would have happened had you gone against chemotherapy and maintenance of durvalumab?
It probably would have been a little better, but these were patients that were felt not to be candidates for it.
So do you think that this PARP inhibition has a story in the future in bladder cancer, or is this something that we can park and say we learned something great in prostate, great in ovarian, but not for us in bladder cancer?
So I think there is potentially a role in maintenance therapy, adding PARP inhibition, and there's the UK study did that without immunotherapy and showed a benefit in selected patients. So I think that's where we need to go. I think that would be a good place to plant, assuming that maintenance therapy sticks around. If EV-Pembro blows out in maintenance immunotherapy, that could be a problem.
Any questions from the panel to try and torpedo Jonathan's otherwise excellent answers so far? So we're going to vote on Jonathan's answer. Green, he's definitely eliminated. Okay, we're good to go with the next question. We've got 3 candidates remaining.
It's getting harder. These are going to get harder too.
Okay, are we good to go? 5, 4, 3, 2, 1.
All right. Matthew.
All right, so this is RC48. This is disitamab vedotin. It's an antibody drug conjugate directed against HER2. The antibody is a different antibody than trastuzumab, binds to a different domain on HER2. And it's conjugated to vedotin, which is the same payload as enfortumab vedotin. This was a study done in the UK. This was actually the combination of 2 phase 2 studies with similar eligibility criteria, looking at this drug as a single agent in patients who had at least 1 prior line of treatment. In this analysis was limited to patients who had HER2 overexpression at the 2+ or 3+ level, or who were FISH positive. And so you can see the response rate here is quite respectable in a heavily pretreated patient population, 50.5%, pretty reminiscent to what we see with enfortumab vedotin, and that there does seem to be an enrichment in the likelihood of response based on higher levels of HER2 expression, with the IHC2+ FISH positive or IHC3+ with a response rate of 62%.
Matt, are all of the, what's the toxicity profile of this agent? And is it the same as EV because the payload is the same?
Yeah, so it's similar from a payload perspective, but doesn't have some of the side effects that might be more driven by the targeting, right? So less skin toxicity because of the lack of nectin expression, because nectin is highly expressed in the skin. There's hypertriglyceridemia, which is a little bit different than with enfortumab vedotin. A little bit more cytopenias. Neuropathy seems to be similar to maybe a little bit less.
Matt, do you think it'll get developed in a subset of patients, like towards the top of the slide, or just in all comers?
So the trial that was also presented at ASCO this year was disitamab vedotin plus toloparalimab, which is an anti-PD-1 antibody. And the response rate to the combination was really quite high, similar to what's seen with EV plus Pembro. And when you look at the HER2 3+ population, the response, it's small numbers, but the response rate was like 90%. Yeah. So I think, in select patients, that might be the go-to regimen.
Matt, do you see a role in HER2 plus 1?
So in HER2 1+ tumors, there was a separate abstract presented at ASCO showing activity. The theory behind that is that this is a cleavable linker, and so you don't need so much expression to show activity.
There's a question from the audience.
So two questions. One is what proportion meets the inclusion criteria, and what were the two? Because like FGFR3, you see responses, but they're quite short, aren't they? So were the responses of worthwhile length as well as?
Yeah, duration of response wasn't presented, and then in terms of the proportion of patients with urothelial cancer who are HER2 2+, 3+, it depends on the study that you read and whether or not it's gastric versus breast cancer scoring that's used, but it's probably around 30 to 40%.
Matt, you did the discussion, I think, at ASCO GU and of a number of trials, and you talked about combining them with immune therapy, and you sort of made a summary to say that you thought some drugs combine better than others, potentially with immune therapy. Do you want to rehearse what you said at that meeting?
Yeah, this came up with Chris's question before that. Is there a difference when you combine ADCs with IO? The difference between that and what we've experienced with chemotherapy, the platinum-based chemotherapy, and then is there a difference within the different ADCs? In small data sets, we basically have 4 trials, small data sets, but there does seem to be a signal. With vedotin, the response rates are really high. With topoisomerase inhibitors, they're a bit lower.
But in different therapy settings.
In different settings.
Question from the front, Dan. Could we give him? You're far away, Nick. You can run past it better. That's fantastic.
Aren't you the guy that was trashing subgroups?
Those were subgroup analyses looking at effect sizes in randomized studies. I think it's a different.
This is hypothesis generated.
Yeah.
Hypothesis generated. Yeah.
Okay. Any questions from the audience on RC48? Matt, last question for you. Enfortumab vedotin seems to be successful. We talked about a lot of frontline trials, and we talked about metastatic trials. We didn't see RC48 appear prominently in the future, and yet you're saying you think this is an active drug that combines well with therapy. What is a development plan, or what development plans can you share with us in the meeting today that's in the public domain around the future of this drug?
So this is a drug that's been developed in Asia and with multiple phases. It actually has accelerated approval in Asia in gastric cancer, if I remember correctly. And so all the development's been done in Asia. Now it's being developed in the United States. There's a large phase 2 trial mimicking some of these cohorts, including combination with Pembro, and then a phase 3 launching as well. So the reason I think it didn't come up before was that the development is way behind EV, but its phase 3 trials are planned.
Sweeney.
What do we know about cross-resistance between the different payloads? There is activity of SG after EV, but how much is that going to be viable?
Yeah, I mean, it's all anecdotal, but anecdotally we do see patients respond. Some patients respond going from one to the other.
We don't know the mechanisms of resistance to EV, for example, and so I think, if it's target-based, then it'll probably be active. If it's the cytotoxic based, it's going to...
Jonathan's been eliminated, but he's helping Matt in his question. I don't know if Jonathan should be put back into the competition for cheating, which I haven't actually thought about, but he's in danger of being back in the competition. We're going to have to think about that carefully, Jonathan, but you're certainly on very thin ice there. And we're going to vote for Matt right now. Let's have a look and see where we are with Matt.
I just need to clarify, is it bad or good to be voted off? Is it bad or good to be voted off, Tom?
Is it good to be? I'd want to stay in probably, but there we go. Okay, here we go. So we've got 3 left. We're good to go.
Actually, Tom, I jeopardize myself here, but there is very early data, minimal, that actually loss of Nectin-4 may be part of the mechanism of resistance. We don't have concrete data yet.
I think we should put Jonathan back in for cheating. Is that fair? So Jonathan, you're back in the competition. Here we go. So 3 to go. 3 left. 3 questions left. Shilpa, far away.
Did you mean to?
No, I didn't mean to.
Rules are rules. You buzzed in. And this is a Tom graphic slide, so good luck.
Shilpa, did you not mean to buzz in?
It's too bad she buzzed.
I can be a little
Far away, far away, far away.
It's a hot buzzer, Tom.
It's a hot buzzer.
I think this is where we stand with the biomarkers for immunotherapy in urothelial cancer, and the short answer is we still don't know what the right biomarker is.
Do you think these colours are fair? Do you think giving red to PD-L1 and giving bright green to ctDNA and T effector signatures is fair? Do you think, this is one of my slides, do you think I've got this right, or do you think I've got my colours wrong? I think the length of the slides is right in terms of the robustness of the data. The question I've got is, do you agree with my colour codes, or which would you change?
I love the color code.
Or are they all going to turn red as they lengthen, right?
Will they ever lengthen?
Is it a science like or an aesthetic like?
is that denoting something? So okay, let me see that. Less promising. So I think for the PDL1, the red and the pink, I don't agree with the pink. I don't think you can really tease out between tumor cell and the immune cell at this point. So I think both would be red. Tumor mutational burden would be green, dark green rather. The light and the T factor signature is also dark green. TGF beta is yellow is good and ctDNA green. I agree.
Opening it to the audience. Any questions on the biomarkers or from the panel on the audience, in the knowledge that if you are seen to be helping, there is a chance you might be put back into the competition, as was the case with Jonathan. So I've got 2 questions for you, Shilpa. The first is, yes, sorry, I'm terribly sorry. Rana,
Dan and I were just chatting that really what this means is the more you study a biomarker, the less.
Do you want to have one of these? Because I think.
The less. I can't catch. Okay, there we go. But the more you study a biomarker and the more robust data you get, the less enthused and more tempered you are about
Everything's
its ability.
Until it's not.
Exactly. So everything kind of in the, first part of the curve, the shorter the line is, the more excited you are. There's less data to show you that, yeah, it's going to fail.
I think that's right. Shilpa, which of those do you think are we going to get a green coming to positive randomized phase 3? Are any of them going to, is Rana absolutely right, and all that happens is the longer they get, the redder they become? Or are any of those promising ones going to end up changing practice?
I think the tumor mutational burden is the most promising.
Yes.
As far as I think, because we have looked at it in the real world and showed that patients with high tumor mutational burden respond better to immunotherapy.
And what about ctDNA in the IMvigor011 trial?
I think that has potential to become positive. I would still watch out for it.
Okay, any more questions from the audience, or we're going to vote on Shilpa's slide? We're going to vote.
It's actually your slide, but yeah.
Well, my slide. Sheep's interpretation of my slide. Okay.
Good job.
Okay, we're going to vote. We've got
Good
green
job.
again.
Yeah.
So we've got 2 left in the competition. We've got Jonathan,
That's
for
right.
obvious reasons. Oh, I thought you did rather well. Here we go. Kala, you good to go?
Sure.
5, 4,
Do I
3,
need to-
2, 1.
I think it was Kala.
That's me.
By a nose.
So looking at VEGF-targeted therapies in bladder, I mean, I think there's been a lot of enthusiasm for VEGF-targeted just from the renal world, from everywhere else. Pathologically, there may be some rationale, but we've had a whole bunch of negative studies, as you've seen there. I don't know if my sorafenib study, which is probably one of the most negatives there.
I apologize, Carla. I
It's
realize.
so negative.
So
I
bad.
mean, hello.
So negative it didn't make the slide.
It didn't make the cut. But yeah, I haven't been that enthused about VEGF-targeted therapy. And when we look at LEAP-11, again, same thing. I mean, and then we talked about this study already before. So I'm not sure why it's not panning out, but it's just not there. Now, combination study, Shilpa's study of maintenance of alemab plus cabo, there could be something going on there. I think single agent VEGF has not really been that exciting for me.
And also the Montis trial, I think at recent ASCO GU, it was cabozantinib different, but it has a VEGF activity, also not a good signal there as in the first line setting.
Earlier, we showed a neoadjuvant chemotherapy plus VEGF targeted study, which looked quite positive. What is that? Just a black swan event, or is that drugs working differently in different settings, or is that how do you square that?
Yeah, I mean, I think there's a bit of that. I just don't know that maybe there's also this issue of there's a lot of redundancy, and so going after one VEGF pathway isn't getting us where we want to be. For me, it's just not working. But Jonathan, you did the study with.
Be careful, Jonathan.
I can't.
I'm still in, so it doesn't matter.
He's still in.
I think I don't think we understand what nintedanib is totally targeting, right? We know it's targeting FGFR, but it's not just FGFR, and it was pretty compelling data. So it needs to be repeated. Targeting VEGF narrowly clearly doesn't do anything. Whether in the maintenance setting it's going to be different, we'll find out, and we'll see how that goes. That's Shilpa's study.
Shilpa, do you want to defend that study? I mean, people are saying VEGF, and I know you're tweeting, and I'm sorry to interrupt you tweeting, but people are saying, gee, we're not that excited about VEGF, and sorafenib's a dirty drug, and so they're all multi-targeted.
I think the beauty of that study is that is in the maintenance setting, we've already selected out the good players and we're trying to intensify belimumab backbone. And Tom's study from maintenance cabozantinib single agent didn't show much activity, but it did show a long duration of response. So I think banking on that aspect and combining with avelumab, we want to see if it can outdo avelumab.
Fair. So Kala, so Jonathan, it's over to you. One slide to go. You don't have to buzz in. Right. You don't have to buzz in.
It's so much fun, though.
So fun.
Okay, here we go.
Back to medical job today. Have to tell you a story.
Okay, so
you got to buzz, Jonathan. You got to buzz. That's part of the game.
So here we see the data that led to the accelerated approval of sacituzumab govitecan in advanced urothelial cancer. The response rate, 27% in a multiply refractory patient population. A very small minority of patients had prior EV. You can see that there's a lot of about half the patients had 3 or more lines of therapy. We're waiting for the randomized phase 3 trial for full approval. I personally find the drug very useful. It's largely well tolerated in patients, and there is anecdotal activity, I would say, in post-EV patients. In my practice, either minor, they're not always great responses, but they're often durable beyond what I might expect if I'm giving a taxane or some other agent. And so patients actually tolerate this well, and if we ever do a randomized trial, I think that quality of life is actually pretty good with this drug for
Where do you
patients.
use it in your practice?
So I'm looking at patients who are post-EV, and I'm also looking at patients who have neuropathy and giving it to them sacituzumab beforehand. There is level 1 evidence versus accelerated approval, and so I'm tending to prioritize EV at the moment. But I've been impressed at how reasonably easy it is to give compared to what I might have expected before I started using it.
What do you think about the neutropenia, Jonathan?
A lot of it is asymptomatic and not concerning. There are some patients who need dose reductions. I probably violate the ASCO guidelines more than I should with giving growth factor support.
Do you violate into giving more or less? When you say you violate the guidelines, you're giving more
I'm giving
GCSF?
it as primary prophylaxis for people who've had bone marrow toxicity from prior lines of therapy where they've needed growth factor support before. And so I'm using it in a sort of secondary primary setting.
Petros, I promise not to bring you back into the competition. But I know you've been very involved in the sacituzumab program. I'd like you to talk about 2 things. Firstly, I'd like you to talk about the neutropenia issue and who should be getting GCSF, because I think it's an important point. And then the second thing is about how it's combining with other immune therapies.
Great question. So actually, I think, Jonathan, you're doing a good thing and you do not violate ASCO guidelines. If you look at the guidelines, most of the patients we treat meet many of those criteria. So I have adopted practice, Tom, that I'm using as a primary prophylaxis GCSF, and I'm doing a daily GCSF, day 2 to 6, so to preserve day 8 intensity, and then 9 to 15. It's, of course, extra injections for the patients. But with that strategy, I have done well keeping the day 8. Does it matter for long-term outcomes? We don't know for sure, but there's some early data that those may matter. So I'm using that. I have actually much less of a problem with neutropenia with that primary GCSF. In terms of the combination with IO, we saw the data from ASCO GU 2022, combination between SACH and pembrolizumab in platinum refractory disease, a difficult to treat population with very low response rate and short-lived responses to platinum. We saw a response rate at that study 34%, second line setting, about 6% disease control rate. We have longer follow-up, and we're going to show the data very soon.
Matt, question for you. 27% looks less than 40%. 40%
yes.
EV. Yeah.
My answer is yes.
So EV's been consistent at 40%. Never been, always, sometimes a bit higher, but very consistent. 27% seems lower than that. Do you think this drug is as active as EV? And are you as confident that the randomized phase 3 immune and platinum refractory trial is going to be positive as EV was?
I don't think it's as active, but similar to Jonathan, I've been impressed with the durations of response that I've seen even post-EV in a subset of patients. So I think it's likely to be positive, but I don't think it's a slam dunk.
Anyone, I can see, Dan, you've got your hand up and you're grabbing that box really tightly. So it suggests you've got something you'd like to add. Fire away.
I'm curious, as a registration path, Is this the way you see bladder cancer going forward with new drugs, that we start with single arm, 100 patient experience to get accelerated approval? Is that door closed now, or is it still open to others? Number one. Number two, I'm curious, this is roughly 30 responders. What's the confidence interval around that? And with a targeted therapy like this, I mean, how confident are you that this really represents a broad patient population?
Well, so TROP2 is expressed very broadly in urothelial cancer. So I'm less concerned about that question. I don't actually know that 95% confidence intervals around that, but it's probably reasonably not that huge. What I think is that, people are there's the rush to move everything first in the disease. It's happening in all of our cancers we treat, and so earlier is better for patients, but as a way to develop new drugs, it's very difficult. Matt has talked about using ctDNA clearance in the adjuvant setting as a way to develop new agents, but it's hard to do that without some evidence of real activity of the drug. And so I actually think that we are now seeing 5th and 6th line therapy, 7th, 8th, 9th line therapy in bladder cancer. We did not have that. It was an exceptional patient, and now it's something I'm struggling daily with, trying to find trials for my patients, because they progressed on EV and SG and immunotherapy and this other trial and that other trial. And they're looking at me with PS1 and good organ function. What do we do next, doc? I'm like scratching my head. I'm not sure yet. We'll figure out what comes around.
Petros?
I agree 100% with Jonathan. We see more and more of those patients in the clinic, which is a good thing. People live longer. Many of them, it's now more feasible to give a 4th or 5th line of therapy, which is great. At the same time, we have this question about sequence of therapies. I have seen anecdotally responses to SACHI after enfortumab. In that trial, there were 10 patients who had prior enfortumab, and they had partial, 3 of them, 30%, whatever it means, had response to SACHI. The other point I have is we're going to stay tuned. We're going to have longer follow-up on that cohort and the cohort 3 with pembro plus SACHI coming up soon. In terms of the registrational pathway, I think there's still a way to do that approach. It makes sense for the companies, of course, and it's the way in. And of course, if they get an accelerated approval, they have more resources to do more studies. I'm okay with that. And at the end of the day, it's more options for the patients.
I agree with that. Just the question is, though, what's the bar, right? Because you said 40%, now it's 27%. What's your bar for the next drug that comes along for this space?
Dan, you raise a great question. It depends on the setting. If you go post-EV, now the benchmark is about that. It's about 27, 30%. That's a benchmark post-EV. Now, if you go post-SACHI, right, your benchmark is docetaxel, assuming the TROPiCS-04 will be positive.
But then also remember that EV pembro may move much earlier. So the whole thing is going to change. It's kind of like a train station in Europe, like everything is going to move along.
Petros, I've got a question for you. You presented some data with sacituzumab, which has an SN38 payload with immune checkpoint inhibition, and the response rate was it 36?
34%.
34%. 34% doesn't sound as high as Jonathan's 70 or 65% with, yeah, 65. There was a 71 kicking around there, but yeah, sorry, no.
I agree.
I'm good at numbers. Petros, why have you only got 34% compared to the 65% with sacituzumab? Is that just statistical noise, or are these drugs combining differently? And if it is different, why?
A lot to answer here. I'll try to be brief. Number one, different therapy setting. Probably Matt Galsky, that's why he asked. We have a first-line statin-eligible chemo-naive patients versus a platinum refractory population. Just for context, in the cohort 3 with a combination of pembro plus SACHI, about half of the patients had platinum first line. The response rate was only 15%. And median time to set SACHI pembro was very short. Tells me that the population is very hard and very different than the pembro EV population. The second point is the biological plausibility and the mechanism. There are some data suggesting that the topoisomerase 1 inhibition may upregulate PD-L1, may upregulate antigen presentation. It makes sense biologically to combine with IO. On the other side, there are people who say, oh, you may deplete lymphocytes, and is that synergistic or not? It's hard to say. I think you can argue either way. I think with longer follow-up, we'll see a little bit more about the response rate, duration of response with pembro SACHI, and of course the signal of PFS-OS.
We've not heard about a frontline randomized trial with sacituzumab yet. We obviously have them with EV. Why is that? And if there was a trial, what would it be?
So there is an ongoing Trophy U01 trial, has multiple cohorts. Cohort 4, we're doing a combination of sacituzumab plus cisplatin, and then maintenance of saci plus avelumab. Try to get some data in the frontline setting, especially with different.
So this drug is in the frontline setting?
Not in a randomized setting.
Correct. Single arm study, not randomized. But the question there is, can you potentially, in theory, displace gemcitabine with such a more active drug in theory and combine it with cisplatin with non-overlapping toxicities to some degree?
I have a question. Can I ask a question?
Yes,
sure.
please.
So, non-overlapping toxicities, myelosuppression with cisplatin and sacituzumab, the febrile neutropenia is a black box warning for sacituzumab, the fatalities. So what are your thoughts of that combination frontline?
Great point. Even more of a reason to give growth factor in those patients and give it as a primary prophylaxis. And again, my strategy has been day 2 to 6 and 9 to 15. That strategy has done well anecdotally, and I think it makes more sense in the context of the trial.
How about combining it with EV-Pembro instead?
It's an interesting question. I think there is a study, I think Guru Sompavde is doing a study, frontline, we have to say. Guru is doing a study with Saci plus and Fortumab in pretreated disease. We have not seen any data yet, but this will give us some safety signals, of course.